Thank you very much, Yusuke, for agreeing to give a Build a Cell seminar, and you can get started.
Yeah, thank you so much for thank you, Kaito, for giving this opportunity to present my recent works about the astral cell project. So my talk today is this title, Active Cytoskeleton in a Cell Size Space, that Web and Motility. Okay. Oh, sorry. So before going to the detail of my talk, I would like to introduce myself. So my name is Yusuke Maeda. Now I'm an associate professor at the Department of Physics at Kyushu University. And Kyushu is a south area of Japan, around here. And I spent 9 years at this department to work on some physics on the biology of some intercellular dynamics. So the Kyushu University, its layout is like this. It's a one-dimensional campus. And this university is close to the Japan Sea coastal side. So we can enjoy fishing and getting fish and also doing some barbecue inside of the campus. So this is a very nice place to work on the academic research and enjoy the countryside of the Japanese life. But right now, our laboratory is a transition period. So because my laboratory will move to the Kyoto University for the coming April, so after 9 years of the Kyushu University, we will be started a new journey to the Kyoto University. So in that case, this seminar is very nice opportunity for me. Just think about what I have done in Kyushu and then what we should do in new place. Okay. So central motivation for me is to do research is to find a very fundamental rule between some materials and biological systems. So biological systems are made of a very large number of the molecules and some associated ions, small molecules. So it's infinitely complex systems. So to bridge some gap between some material and the biological system is one of the very big challenge in the modern physical science. So, but to bridge such a gap, of course, more recently, few decades, there are several physical science has been widely developed. So one, let's say, growing field is a soft matter physics. For instance, soft matter is some flexible material that shows a big deformation under the small, let's say, mechanical perturbation. So soft matter is kind of some polymers and membranes and liquid crystals. So such materials surrounded in our living, let's say, everyday life is also some, let's say, focus of the modern physical science. But the soft matter is somehow the big limitation to understand the biological systems. Because it's a very simple bubbles and membrane, sometimes it do not have a catalytic activity. So for instance, the protein enzyme has a catalytic activity and consider the chemical energy and make some products for the new materials. So such kind of some functional activity, chemical activity is necessary to bridge some gap between the material and the biological systems. So to bridge the gap, our laboratory is now focusing on the active matter system and also to understand the biological systems. Okay, so active matter is some material that consumes chemical energy and convert it to the mechanical action. So it's a new class of the autonomous and non-equilibrium system. So active matter is quite a wide class of the materials from the nanomotor or the molecular motor proteins like kinesin motor protein and microtubule gliding microtubules. And at the micron scale, the group of the epithelial cells is classified into the group of the active matter. So this kind of autonomous active system has a very nice emergent spatial dynamics, this kind of corrective motion and pattern formation. So to understand the active matter system is one of the very big challenge in the modern kind of a soft and active matter physics research field. Sorry, okay. And the active matter, it's not, sorry, I would say, sorry, the active matter is not, let's say, limited in the some natural world. So active matter is also present inside of cells. So one famous example, one representative example of the molecular, let's say, active matter system is this kind of oxygen filament and the myosin molecular motor system. This myosin molecular motor is an enzymatic protein that consumes the chemical energy, and that energy is converted into the mechanical action to move out this oxygen filaments. So this myosin molecular motor make a complex with this oxygen filaments and make us some cross-linking structure and make us some gel-like complex like this. So this, let's say, complex is known as actomyosin gel. So it's generate a contractile force spontaneously and make this kind of contraction of the molecular gel composite. So this is this actomyosin active cytoskeleton system is also present inside the cell. That regulates a lot of the cellular dynamics confined in the cell size space. Here is a very famous example about how the actomyosin, the cytoskeleton system regulates the cellular dynamics. The first one is the organelle positioning that is regulated by the one kind of the myosin molecular motor. So the one type of the rodent egg must be precisely positioned this nucleus and the spindle structure at the early phase of the fertilization. That this spindle structure must be present at the center, but after the maturation of the egg, this spindle must go to the edge of the membrane boundary. So this kind of positioning must be precisely regulated for the normal fertilization. And the second example is, of course, everyone may know, the cell migration driven by the actomyosin. So the cell division, to drive a proper cell division, the actomyosin makes a ring-like structure and make a contractile stress like this. So this kind of a rich dynamics of the actomyosin is regulated simply just one set of the molecular components, just oxygen cytoskeleton and molecular motor. But one interesting question for the physicists should be how does this kind of a single set of the actomyosin just organize a rich order dynamics in the intercellular space? So to answer that question, we just need to think about the synthetic biology approach, because the living cells have a very complex signaling cascade inside of a cell, so regulates the cytoskeleton and the molecular motor protein itself. So to make some model system with reduced complexity, we decided to build the artificial cell model. This is a conceptual strategy for how to make complex cellular environment in the model system. First, we just, let's say, separate or extract the very important molecular components from the living cells. And those important molecular components is encapsulated in the artificial cell size space. And then we just put the molecular visualization die inside of this synthetic minimal cell. and try to observe its dynamics under fluorescent microscope setup. So by observing the emergent dynamics of this synthetic minimal cells, we can understand its, let's say, underlying physical mechanisms of some active cytoskeleton. So this model system has several advantages for the physics study. So first one is, this is a well-defined model system. We can apply the active matter physics or nonlinear dynamics to understand its observed experimental, let's say, dynamics. So, and for the model system, advantage is we can control its cell size by changing the, for instance, diameter of the cell confined space. And also we can manipulate the, for instance, the molecular components by putting the purify the, let's say, the protein inside this artificial cells. We can, let's say, control the internal, let's say, for instance, the mass contractility or other nucleation factors, and so on. So such kind of manipulation of the physical length scale and the manipulation of the chemical state, those two, let's say, manipulation of the molecular control can be possible in this, let's say, the artificial cell system. Okay, so this kind of course, the artificial cell approach has a very long history, almost more than 10 years ago. So but I think this artificial cell construction should be one of the most developed experimental approach to study the cellular dynamics in the in vitro system. So the shown here is our actual experimental setup to build the active cytoskeleton in a cell size space. So, sorry, I'm now talking about my talk in my living, but my wife is breeding the rabbits, and sorry, rabbits is very excited in my talk. Sorry, sometimes it's very noisy. Anyway, so this is actual experimental setup. So this is we just extract actin cytoskeleton and the molecular motor from the frog egg, and it's this physiological concentration of this frog egg extract for the actomycin system that is supplied from my long-term collaborator, Professor Miyazaki Makito. He is now Riken VDR. Okay. Okay, so and then this extract is confined in let's say the mineral oil absorbing the lipid phosphoglycerine, and this method also developed by the Professor Miyazaki. So we just let's say start to collaborate to work on this kind of project. So we just make an encapsulation in the extract in oil system. So this is a mere, let's say, just a simple emulsion system. We have, let's say, not many types of the emulsion, not many types. This let's say, same extract concentration, but the size of the droplets is somehow different. So then by express after the encapsulation of this extract in this lipid monolayer emulsion, we put this let's say artificial cell droplet under this glass slides, and so to observe this actomycin dynamics under fluorescent microscopy. So the action dynamics is visualized with TMR life act. So this is a very nice peptide marker for the filamentous action, and it do not interfere with let's say depolymerization dynamics. So we can expect a very nice turnover rate of this F action let's say visualization system. So then we just put this kind of cylindrical shape of the actual cell. It's not a spherical shape. So as one notice, it's not a spherical one. It's a cylindrical shape, and the aspect ratio is about 0.3 to 0.6. I think the reason why we prefer the cylindrical shape is, yeah, spherical shape is still complicated for us. The created two-dimensional space is much familiar for the some numerical simulation I will show later at active fluid dynamics. So, and also cylindrical shape is sometimes better for the visualization. So that we make this squeezing, let's say, geometry of the artificial cell. Okay. So sorry, it's very long introduction. So yeah, here is a typical example of what we have observed in our experimental system. So here, this one is a top view of what we found in the actual cells. The yellow color is a filamentous action, so visualized by the TML life act. So just a few seconds after the encapsulation, we can see the big contraction of the actin gel, and then you will see the periodic wave formation from the boundary to the center. So this ring shape is also the actin site skeleton. Let's say this is a contractile force is generated from the mouse activity. I will show it later. So the right one is a bright field image. You can do the PIV analysis to visualize the flow dynamics, but in this talk we can skip this PIV analysis just to focus on the fluorescent, let's say, microscopy dynamics. Okay, so very interesting dynamics. The periodic wave of the oxygen cytoskeleton can be occurred in just an encapsulation in the cell size space. Okay, so this is a time graph representation of what we found in the actual cell system. Okay, this axis is time and the horizontal axis is space. So the from here is some boundary of this droplet. So you will see the oxygen wave has after the encapsulation, big contraction occurs, and here is some big cluster formation of the active gel. And then from the boundary, the ring-like shape generated, and it's contracted due to some Martian molecular motor contractile force activity, and the ring go to the diameter shrink. Okay, so we can define the velocity of the actin wave from this kind of graph. And also our droplet has a main size of the variety of the diameter. So we can analyze the different size of droplet and what kind of a conductor velocity can be found in each droplet. So interestingly, we can see the proportional relation of the velocity as a function of the droplet size. So this means that just a bigger droplet has a large amount of some oxygen and myosin. So a large amount of the proteins generates a larger initial, let's say, the contractile velocity. Yeah, that's it. So, but what is quite interesting for us is the wave period. Wave period means some time between the first wave and the second wave and third wave. So the time difference, let's say this time difference, it's almost constant or weekly, let's say, proportional to the droplet size. So this means I think this wave period of time it's mainly decided by the maturation for the actin meshwork growth. Okay, so this is so that this actin and this our actin and myosin extract also has other types of the associated proteins like nucleation factor of the actin polymerization. One very famous major nucleation factor is APT3 inhibitor, sorry, APT3 complex. that, let's say, activates the BASP family pathway to nucleate the actin filament very close to the membrane boundary. So let's say, check this kind of nucleation factor is really involved in our actin wave formation. We just put in molecular perturbation to add CK666 to inhibit this upstream regulation, upstream complex. So by adding this upstream complex, we can see that some wave it's somehow fluctuating. So the shape of the cluster is not so stable. Also, the contraction wave velocity becomes slightly, let's say, larger. So slow wave velocity, the faster, slightly faster wave velocity. So, and also the mousing also involved in the contractile dynamics, shrinking dynamics. So by activating the mousing and let's say the inhibiting the mousing activity, we can let's say see the increase of the mousing activity, sorry, the wave velocity, and reduce the wave velocity as well. So the from those molecular perturbation experiments, we can say the major factor involved in this actin-myosin ring formation is one is up to 3 nucleation factor, and another driving force is a myosin molecular motor. Okay, so from those experimental results, we just show the build proposal this kind of simple let's say model of the optimization wave formation. So that's an absolute free complex is activated at the repeater interface. So and it makes a dense network of the actin filaments crosses the membrane boundary. So once the density of the actin network becomes larger, myosin molecular motor can bind to this actin meshwork and generates a contractile stress. And then if the much more mouse molecular motor attached to this dense actin meshwork, contract stress bigger and becomes bigger and bigger. And then once this contract stress exceeds some threshold value, this cortex will detach from the boundary. So it will generate a wave propagation toward the center. So this is a very simple mechanism to explain why we can see that this kind of a wave propagation can be found in our artificial cells model system. Okay. Okay. So I think sorry, Kate, we should answer the question in the chat.
So most of the time people choose to answer after the talk. So
After the
people
talk, okay.
will leave the questions and then you can answer later.
Yes. Okay, thank you.
Thanks.
So yes, please leave a question if you have some. I can answer after my talk. Please leave a question any point. Okay. So this is some very interesting wave formation in the confined cell space. But we find a very interesting transition from the kind of continuous flow to the periodic wave formation. So when actually we just control the machine contractility by adding the let's say curriculum A as actin-myosin activator. So by changing this curriculum A myosin contractility, we can see the periodic wave formation as shown in the previous slides. But at a lower myosin contractility, so in that case, what we can see is only the continuous flow from the membrane to the boundary. So this kind of a continuous flow has been reported in the several previous studies, but we don't know how this kind of a flow to wave transition can be possible by simply changing the myosin contractility or other associated parameters. So here's a time graph, steady flow and a periodic wave. Here's So yeah, just by changing let's say massing conductivity, we can see the wave to flow transition can be controlled. Also that by let's say partial activation of the massing conductivity has found it's like a base sharp transition. So the continuous flow to the periodic wave has a sharp transition like this. Okay, so to answer the what kind of a physical mechanism of periodic action wave, and is there any biological relevance of this periodic action wave? To answer those two questions, we decided to build some numerical simulation model of the active cytoskeleton in a confined space. So this is a simple schematic model of our active fluid model. So actually, it's just describing the stress term of the actomycin fluid. So in the case, in the actin and the myosin fluid, just let's say made from the three components, so actin proteins and also myosin molecular motor and the other water fluid solvent. So if we just only consider this kind of protein solution, what we have to think about is a passive viscous stress, like bulk viscosity and shear viscosity, like this. And but in our active fluid model has a fourth generation material that is a myosin molecular motor. So we have to think about its active contractile stress. So this active contrast stress is supposed to have this kind of a relation. Its stress is proportional to the ATP chemical energy difference. So that means it's also related to the catalytic activity of the myosin molecular motor. And this is the density dependent force of the molecular motors. So this has an arbitrary functional form, but in this study, we let's assume that this density dependent force is a kind of sigmoidal function, like a Hill, let's say Hill equation. So by let's say considering that those two stress components, we can describe them. Let's say what kind of internal stress can be described by this active fluid model. Okay. So actually, this active fluid model is widely developed in the active matter physics field, but this we just let's say we just try to take over those two powering papers about active fluid model of the actin cytoskeleton system. Those two papers are very good. So and then by considering those two, let's say, internal stress of the active fluid model, we just solved numerically this let's say liquid solid fluid model. But to describe the kinetic equation, we only consider two physical equations. First one is a force balance equation like this. We just consider the divergence of the stress term, and also this force can be balanced with viscous stress, viscous friction term. And also second one is a mass conservation. We just simply think about the advection by the actomycin gel and the diffusion, action polymerization, and the depolymerization rate. So to numerically solve those two equations, we let's say let's use a phase field model approach. By taking a non-dimensional parameterization, we can get some flow field equation and a density equation as shown here. But one very important non-dimensional parameter is a Peckel number as shown here. This is a non-dimensional parameter as a ratio of the active stress to the thermal diffusion term due to just thermal fluctuation. So the larger Peckel number, let's say, means contractile stress is very large, and that can overcome the thermal diffusion of the molecular components. So then let's say, make a simulation inside with the cellular confined space, we consider the circular boundary like this. This is defining the boundary condition like this. So but our boundary has kind of a nucleation activity, so that induce much dense, let's say, axial meshwork. So we consider the surface polymerization rate is much higher, 10 times larger than the bulk polymerization rate. So by doing this boundary condition, we numerically solve this phase field model and obtain this kind of result. So at a lower Peckel number, the Peckel number is about 30. So we can see the continuous flow from the boundary to the center. So as seen in the experiment. And by increasing the furthermore, Peckel number is closer to 100, we can see the periodic action wave is reproduced in this our numerical simulation model. So I think this is a very nice example. The contractility itself can regulate the spatial temporal dynamics in the confined cell size space. And by thinking of this numerical simulation, we can find by increasing the contractile stress, the period of the let's say this periodic action wave also slightly increase proportional to the let's say the contractile stress. And also by changing the polymerization rate, we can also find the wave to the flow transition can also be happen. So those two phase diagram tells the effective contractility of the active massing and also polymerization rate. Those two, let's say, the parameters are very important for the transition from the flow to periodic wave. Okay, so this is our numerical model study that very recently, a very interesting paper has been published in the other journal. So transition from the flow to periodic wave also possible with confinement size and also network connectivity of the actin cytoskeleton. So yeah, this result tells, I think, the flow to wave transition can be very universal phenomenon. But I think a control parameter should be several we have a several choice to control this kind of a transition. But I think this kind of a transition or the cell formalization would be very nice model to study about the active cytoskeleton inside with the confinement cell size space. Okay, so this is so far the summary of what we found in the some active periodic wave formation. But I think the audience may interested in this kind of wave is really important for the biological function or dynamics or not. So actually we do not go to the detail about our study, but this kind of acting wave formation is also very important for the positioning symmetry of the organelle inside the cell size space. For instance, this kind of center toward wave formation can be found in the larger droplet size. But if we just reduce the droplet confinement size, we can see the center toward the wave formation is no longer stable. In the smaller droplet size, this kind of a center to the wave is close to some boundary. So this kind of a positioning symmetry breaking can be also found at some interplay between the membrane boundary. If you have been interested in the dispositioning symmetry breaking, please look at this Nature Communications paper. We describe a simple model for why does this symmetry breaking can be happen. But the rest of my talk, I want to show the more interesting, my favorite dynamics are the cell migration or the artificial cell model. I think this is a very nice, let's say, demonstration why that this active cytoskeleton unconfined system is important for the biological dynamics. Okay, so the cell migration is the very famous, let's say, the dynamics of at the cellular scale. But most of the cell migration study is focusing on the adhesion-dependent migration. So cell adhesion also very important for the the focal adhesion is very important for the adhesion-dependent migration. And some epithelial cells make a very strong adhesion to the basal substrate. And then the detaching from the rear edge by generating the large contractile stress. And due to the by consuming the large dissipation in the mechanical energy, these cells can crawl along this onto the two-dimensional plane. But one type of the immune cells and the other type of the cancer cells can move in the confined space, like inside the living tissue, but without any strong adhesion. This is called the adhesion-independent migration or friction-mediated migration. So this migration mode is very efficient for instance, the move inside of the living tissues because they don't use the very strong adhesion to the neighbor network or the cytoskeleton or collagen systems. So it's very interesting migration mode for the one kind of let's say immune cells. So, but to drive this adhesion-independent migration, the actin cortex, that is a dense actin machine meshwork just below the cell membrane, are very important. So when we just think about if one can build a very nice actin cortex in our active artificial cell model, maybe droplets can spontaneously move like this adhesion-independent friction-mediated migration. So to demonstrate this idea, we just decided to add another lipid, PIP2, that promotes upstream dependent actin polymerization just below the membrane. So like this, by adding the PIP2, this actin meshwork can physically contact with membrane boundary. So we can see the strong accumulation of the F-actin molecular component very close to the boundary like this. Okay, so now we can succeed to build the actin cortex in our artificial cell model. So what's happened? And squeezing this droplet glass substrate. So yeah, it's very interesting. So we can start to see this droplet can let's say move spontaneously. So this migration speed is about 1 to 2 micron per minute. And this migration will sustain about 1 hour. So I think this is a very nice demonstration. Actually, this action, let's say, cortex can drive the friction-mediated migration in vitro system. Okay, so to test if it's actually force emission can occur in this droplet system, we put a tracer beads in the outer oil phase. So without some physical contact between the actin meshwork and the lived membrane in the control experiment, the fluorescent tracer particle do not move even under the actin machine wave. So there is no force transmission in the control experiment. But by adding the PIP2 and also make a very nice cortex formation, we can see that this tracer beads is move around some actomation wave. So that is let's say direct demonstration for the force transmission can be happen in this droplet migrating systems. Okay, so and also our droplet system, let's say it appeared some driving force from the physical boundary, and this frictional force from the top and bottom PDN substrate, that is a driving force of this droplet. So, and also this droplet also feel the resistive viscous drag from the surrounding oil. So we can consider force balance equation to, let's say, solve the droplet migration speed from this experimental observation. So because our system is made from the defined factors, so the contractile force is mediated by the frictional force between the contact with the droplet and the physical boundary. So this contractile driving force is proportional to the let's say the contact area between the droplet and the boundary, and the viscous drag is assumed that this is same magnitude of the fluid flow. It's let's say the viscous drag surrounding the cylindrical shape of the radius of the R and height of the H. So by solving this force balance equation, we can get the speed of the droplet migration. Okay, so by solving this equation, we can get the migration speed of droplets. So this velocity has two components. The prefactor, first one is a very simple physical consequence. The alpha V act is a contractile stress, and eta oil is a viscous coefficient. So this is just outcome from the simple Newtonian, let's say, Stokes equation. But we also have another factor, geometry dependent resistance form. So H over D is the height of the confinement size, and D is the diameter of the droplets. So this geometry factor also involved in the migration speed. So this migration, droplet migration is mechanically determined by the surrounding the geometry of the confined space. So from this equation, we can say that if the H over D aspect ratio is small, then in this case, for instance, H over D is small means this artificial cell has a sufficient contact area to, let's say, transmit the frictional force. In this case, droplet can move very fast. But in the larger aspect ratio, in this case, droplet has only small area of the contact between the top and bottom substrate. In this case, the droplet migration speed is very slow, so it has a geometry dependence on the droplet speed. But also, if we can change the height of the confinement size, and even though the same aspect ratio, droplet size is also, let's say, different. So that we have to always consider about the how large the contact area and how large in the confinement size. Those two physical, let's say, length scale can define the migration speed of this friction-mediated migration mode. Okay, so this is an implication to the in vivo situation. This kind of friction-mediated migration is also found in the in vivo kind of immune progenitor cells. Of course, in vivo, let's say, confined cells has a much complex chemical signaling activity, like some mousing activity and the actin cytoskeleton. Also, both kind of activity is regulated by the, let's say, mechanochemical signaling. But similar transition, for instance, the actin wave formation and also kind of deformation And sometimes these cells start to move one direction, like cerebral blood formation. So I think our artificial cell model, it's very nice, let's say, the physical model to think about the mechanical system of optimizing system can, let's say, recapitulate this very complex dynamics of the let's say, confined cellular, let's say, optimizing systems. And is there this kind of transition or the deformation to migration is really decided by the mechanics alone or not? That is, I think, a very important next question. Okay, so this is a kind of a summary of my talk. So we that's a confined active cytoskeleton system in the cellular size space. Under the, let's say, lipid membrane boundary, this optimization system make a periodic action wave formation. And also at a lower conductor stress, it's go to the continuous flow. So this kind of a periodic action wave and continuous flow is very important for the friction-mediated migration. If we impose the membrane interaction between the actin cytoskeleton and the membrane boundary, we can, I think it's a fast demonstration of the friction-mediated migration in the in vitro systems. So our artificial cell model has a much more possibility to study about, for instance, deformation and cell division if we, let's say, try to develop the active cytoskeleton system to regulate, for instance, the polarization and also some partitioning. Such kind of interplay of the chemical signal activity will further pursue how active stress can drive the deformation and cell division in the physical context. Okay, so it's the end of my talk. It's almost the last slide. So let me say actually our actual cell shows a nice transition from flow to wave, and our laboratory is also somehow the transition phase. In coming April, my laboratory we moved to the Kyoto University Department of the Chemical Engineering. So we work on some physics and engineering for the soft and active matter physics, and also self-free synthetic biology and droplet nanomicrofluidics. So for understanding the physical context of the biological systems. So actually, the Kyoto University is a nice cultural capital of the Japan. So it has a beautiful, let's say, kind of a city side and also very nice historic gardens. So if you have an interest in the PhD and the postdoc opportunity in this kind of a cultural place of Japan, please contact me through email or direct mail through the Twitter. Okay, yeah, thank you so much for your attention. And I'd like to thank my long-time collaborator, Professor Miyazaki and Professor Hiraiba, and also my former, let's say, student and postdoc, Ryota Sakamoto and Jan Izuri. Okay, thank you so much. I'm happy to take any questions.
Thank you. Thank you so much. It was an amazing talk. There are questions in chat. If you could read the question out loud before answering, that's going to help with the recording.
Yes, thank you. Very nice question. And yes, I think the first one is that's so sweet. Yes, it's a rabbit, probably in my apartment. Yeah, so her name is Sarasa. Okay, thank you. I will tell it to her. Okay, so second question is, sorry if I miss it. Do you add the ATP in the buffer to keep the action active? Is there ATP regeneration system too? Yes, thank you for a very nice question. Yes, exactly. We have a rich amount of the ATP in our extract. And also ATP regeneration system, we do not add the artificial additive system is not present. Just in our, let's say, extract, we have a lot of the ATP, I think millimolar level. But the duration or the periodicity of the actomycin wave is decided by this ATP amount. So I think our periodic action wave sustain about 1 hour to 2 hours. But if ATP, all the ATP is exhausted, that's stop. So I think we add some ATP and also we add the ATP regeneration system. I think we can expect a much more longer, let's say, periodic wave formation, I guess. Okay, thank you. So, very nice question. And the third question is, do you ever try to stain the phospholipids you use to make these droplets? Okay, yeah, it's also a very nice technical question. Yeah, we have done that, but one big limitation is outer phase also lipid and oil mixture. So we can put the let's point a lipid staining dye, but the contrast is not very good. So I mean, for instance, to visualize the lipid barrier, we can put the how to say the lipid staining dye. That will be sufficient. But in our system, it's an emergent system, so it's not good for the just a fluorescent dye for the lipid, let's say, barrier. So we also tried some fluorescent lipids, but its fluorescent is not very good. And also, we do not have a commercially available for instance, fluorescent PIP2. So right now, the visualization of the lipids are very important technical issue. But it's actually, I think the visualization of the lipids is very important. But we have tried, but not succeeded yet. Okay. Okay, so the third question is, you use a very high concentration of up to 3. If it does, the contraction velocity shows higher than the control. What is the reason for this? Okay, yes. Up to 3 concentration, I see. So we use up to 3 high concentration, but still for our experiment, it's not so very high. I don't know the reason why, but I think our extract has probably physiological concentration. I mean, the very rich density of the efflux filament. Probably we need more up to 3, let's say, factors. And also we need a lot of the concentration of the up to 3 chemical inhibitor, probably. And also the up to 3 let's say inhibition creates a slightly higher contractile velocity. So yeah, it's really important question. Maybe I would show the so maybe I was good to show the phase diagram or the this one. So okay. So actually the polymerase R3 chemical inhibition also change the polymerization rate, slower polymerization rate from the, for instance, flow state. So by adding the R3 complex is like going to the from right in this phase from right to left like this. So in that case, think the situation is a bit complex, but if the action meshwork becomes slightly, let's say, flexible, easy to deform, in that case, I think the wave is easily compressed. So action density is become somehow weaker, slower. In this, but mass in concentration is the same. In that case, the the actual meshwork is more easily deformed. So that's the reason why we can see the shoot, shoot, a bit faster contraction. So that is, I think, the very qualitative explanation why absolute free inhibition becomes a slightly higher wave velocity. But as you can see this phase diagram, the dynamics prediction is not very easy always. So we have to be careful about the concentration of the chemical inhibitor actin and myosin. Okay. So the answer is very long. Um, the next question is a monolayer and bilayer membrane have different polarity. Have you tried to use bilayer of the lipids? Yes. So we have that is our next let's say direction, and probably Ryota Sakamoto, the leading author of this work, is try to work properly, try to work on that project in his postdoc lab. So far, we don't try the bilayer membranes because the bilayer membrane has a very, how to say, the large flexibility and easily compress. So we want to try some much more a simple solid boundary to think about the effect of the intercellular space. So we don't try right now, but I think absolutely the large deformation and the interaction between the actin cytoskeleton and the deformable membrane is a very important project. For instance, to think about deformation and the division. Okay, thank you.
Thank you so much. I saw Mosumi, you unmuted. Do you want to follow up?
Yes. Hi. I wanted to
Yes,
talk with Professor Maeda.
yes, thank you.
Yes. Hello, Professor Maeda. Maybe I don't know you remember or not. We met before in Hokkaido University. So I'm a former PhD student of Professor Kakugo.
Yeah, yes. Yeah,
So nice to meet you here. And
yeah,
I
nice
asked
to
the
meet
third
you again.
question.
Yes.
So I just wanted to ask you something more. So I saw like you change like the parameters like individual one. Have you ever tried like changing two parameters like the inhibitor concentration and also with the myosin concentration, or like two different parameters at the same time?
Yes, it's a very important point. So, so far we don't try the two or three parameter change at the same time. Um, but I think the such a parameter change, it will be very important to think about. For instance, the let's say try to find a new behavior flow to wave transition. But we prefer to let's say check the simple transition by changing the polymerization rate and also changing the effective contractility. But of course, to explore the very huge parameter space, we need to think about the try to explore the changing the lot of the parameter in the huge space. If you have an idea, please let me know. I'm just an old guy, so just try to change a single parameter at a single experiment.
Oh, thank
But
you. Because
anyways, I
that's
think
my
it's
interest nowadays. So I'm working in Professor Alan Leo's lab. We also interested in this type of like cytoskeleton encapsulated synthetic cell system. So yeah,
yes,
that's really interesting. Thank you for your presentation.
yes,
And
okay, yes,
just one adding something more. So she's my colleague. So she wanted
yes,
to
yes,
ask you something.
yeah, please, please.
Yeah, if you don't mind, I know we're over time, but I really very impressed with your work showing the synthetic cell migrating and confinement.
Yes, thank
And
you
I
very much.
was yeah, and I was wondering about the polarity of this movement. Is it
Mhm.
random or can you predict like how it'll polarize? Is it all the same way for each synthetic cell? I
Yes,
was just
yes.
wondering if you can expand on that.
It's right now it's random, but
Mhm.
it can be predicted from the initial dynamics. The
Mhm.
initial I don't know. For instance, at the very beginning, you may see the detachment from the cortex can be happen at the front side of the droplet migration. So
Mhm.
the polarity is built from the initial detachment of the action cortex from the membrane boundary.
Mhm.
So I think once if we can control this kind of detachment of the action cortex from that we can think control the polarity and the direction of the
Mhm.
migration, I think. So yeah, that's initial direction of the most migration and the polarity establishment is right now random.
Mhm.
Yeah, that's next a challenge, technical challenge to control this migration and control the polarity. Yeah,
Yeah.
but you can control you can predict yeah,
Mhm.
you can predict the direction the some polarity direction.
Mhm. Right.
Yeah, from the
Yeah.
very initial, let's say, axon, cytoskeleton, cortex dynamics. Yes.
Mhm. Yeah, very cool. Thank you.
Mm. Yes.
Thank you.
Thank you so much. Thanks for the discussion. I was going to ask a question about the bilayers too, so I'm glad someone
Yes.
did.
Very pleased. Yes.
If there are no more questions, I want to say thank you so much again, and I hope you'll catch some sleep before you have to start the day.
Fine. It's a nice time. I mean, always a mid evening is a nice time to work because the kids and the wife are sleeping.
Yep.
Anyway,
Hey,
so
thank you so much. Thanks everyone, and have a great rest of your day.
okay, yeah, thank you very
Bye.
much for joining this seminar. Thank you.
Bye.